lecture. This is going to be adaptive specific host defenses or immunity. And again, it's our last lecture for spring 2020. This will be really just kind of an introduction. We will only be going through I believe it's slide 19. There's some additional slides, but that information won't be on our final exam. And again, this is just kind of giving you an introduction. If you've had A&P already, you had a really good detailed discussion of host defenses. And if you haven't had A&P, you will get a really good discussion of immunity host defenses. So again, this is just kind of an introduction, and maybe we'll try to just weave in specifically how these adaptive responses will protect us against microbial pathogens, okay? So let's see if we can't go full screen. Okay, folks, so again, this is adaptive specific immunity. It's also been called acquired immunity. If we contrast it to the innate nonspecific defenses, you recall that innate nonspecific defenses provided protection against a wide range of pathogens, right? But as the name suggests specific here, the adaptive specific immune response is going to be specific for usually one pathogen or maybe a specific exotoxin. Another difference is that the adaptive immune response has to be activated. So there's a relatively long lag period that the first time we encounter the pathogen or the toxin, it might take like maybe 10 days, maybe 14 days before, for example, we get maybe a decent antibody response. So this is a huge disadvantage of adaptive specific immunity, is this lag period here. But you'll recall the innate nonspecific defenses, they're always ready to go, right? So the hope would be that if the innate nonspecific defenses can't kill the invading pathogen, they can at least control the numbers and spread long enough so that these big guns, right, Gandalf and the Riders of Rohan can arrive to save the people of Helm's Deep. Okay, getting a little goofy there. All right. There's a huge advantage of adaptive specific immunity, and that is it exhibits memory. We're going to make memory cells, memory lymphocytes. So although the first time we encounter, say, the pathogen or an exotoxin, it's a slow response, it's relatively low response, we don't make a lot of the best antibody IgG, and it's relatively short-lived. But in that first response, the primary immune response, what we're making are these fantastic memory lymphocytes, memory B lymphocytes and T lymphocytes, so that subsequently, the second time we encounter that pathogen or encounter that exotoxin, we're going to have a much faster, stronger, more effective, higher levels of IgG antibody, longer lasting, what we call secondary immune response. And in addition, we're going to make even more memory cells. And we've had an introduction to this, folks, when we were talking about the immune responses to viruses, right? So you might remember we said that in adaptive specific immunity, a huge advantage are production of memory cells. So we'll get that faster, stronger, longer lasting, more effective secondary immune response. So in the adaptive specific immunity, there's two branches, two arms, and the two branches or arms are humoral immunity. Humoral literally means fluid, and if I can get my little cursor going here, so humoral means fluid, and it's because in the body fluids that we find antibodies, also known as immunoglobulins. And in general, folks, we're going to say that humoral immunity provided by antibodies or immunoglobulins, will protect us against extracellular pathogens and toxins before they get inside our cells. Antibodies can't cross cell membranes, so usually once a toxin or once a pathogen gets inside our cells, the antibodies can no longer help us. And if we wanted to ask which cell makes antibodies, we're going to see it's our B lymphocyte, sometimes called B cells. B is for bursa fabricus, a lymphoid tissue found in birds. We don't have a bursa fabricus, but we're it's been discovered that our B lymphocytes usually mature in the bone marrow. So we could say B is for bone marrow. And so it's the B lymphocytes that make antibodies. The second arm of adaptive immunity that we'll be discussing is called CMI for cell-mediated immunity. And we'll say this is the arm of adaptive immunity that can protect us against intracellular pathogens, right? So remember, viruses have to get inside our cells to replicate. There are other microbial pathogens that can get inside our cells and replicate. You'll recall those facultative intracellular parasites or pathogens that can live within our phagocytic cells. So CMI is going to help protect us against those intracellular pathogens. In A&P, you've learned it's also the arm of the immune system that deals with cancerous cells, and is also involved in organ or tissue rejection if a patient undergoes a transplant. And we're going to say that we're going to describe the cells involved in CMI are going to be cytotoxic T lymphocytes, which can actually kill our cells that are infected with intracellular parasites. And then we're also going to see, as part of cell-mediated immunity, we're going to see that T helper lymphocytes can activate our macrophages and turn them into angry killers to hopefully be able to kill those facultative intracellular parasites. So if we were meeting face to face, and usually we just have the one 80-minute lecture, I'll start out on the whiteboard drawing the crucial role of a specific type of T lymphocyte called our CD4 positive T helper lymphocytes. They are the most important cell of the immune system. They're like the conductors that are guiding members of an orchestra or a band, with all the different cells of the immune system being the members of the orchestra or the band. And it's through the release of chemical helpers that T helpers will direct all the other cells of the specific adaptive immune response. So without the T helpers, kind of the leader, the rest of the cells don't know how to respond, when it's appropriate, when it's not appropriate. So often the T helpers are called the Achilles heel of our immune system. For indeed, if you were to destroy enough T helper lymphocytes, you'll totally cripple our immune system. And this is the great tragedy of HIV. HIV targets our CD4 positive T helper lymphocytes and eventually can destroy enough of them that the entire immune system collapses. And that's when we end up with AIDS, acquired immunodeficiency syndrome. And that usually occurs, I believe it's when our T helper lymphocyte concentration drops below 200 T helpers per microliter of blood. In a normal immunocompetent person, usually your T helper blood concentration is around 800 to 1200 T helpers per microliter of blood. So below 200 T helpers per microliter of blood, our immune system collapses. Right? So this is just a great heartbreak of HIV. And again, folks, what we really need is a vaccine. It would be awesome to have an anti-HIV vaccine. Now, to kind of give the big overview, the big world map of adaptive specific immunity, again, if we were meeting face to face, I would draw this concept map up on the board, emphasizing the key role of the T helper lymphocytes, or CD4 positive cells, in coordinating the responses of all the other cells that are involved in adaptive immunity: B lymphocytes, cytotoxic T lymphocytes, and macrophages. So this, folks, it'll be a little bit harder because usually I'd be writing this up on the board. There is an in-lecture video from fall 2019, and I have to go back and review it to see if it actually shows the board work for this big concept map. But folks, one thing that might help is in our spring 2020 immunology module, there's a PDF of a scan of 2 pages from an immunology textbook. And the second page, you'll see a bunch of cells up at the top, and the figure is labeled figure 57-1, and it says Introduction to the Interactions and Functions of the Major Components of the Immune System. And this, you guys, if I could rearrange it, it would be very similar to the board work I would do. So maybe that diagram will help you. I'll just kind of try to walk our way through this. All right. So folks, if you were drawing this concept map, if we were drawing it in class, what I have you do is draw a little line about halfway down here. And this side we would label humoral immunity. So this is the protection provided by antibodies, immunoglobulins, primarily protection against extracellular pathogens and their toxins. And then over on this side of the page, our concept map, we would put cell-mediated immunity, for it's the cytotoxic T lymphocytes and activation of macrophages that we'll be discussing is as the consequences of activation of CMI. And again, folks, here, central to humoral and cell-mediated immunity is our T helper lymphocyte. Now, the key here is that T helper lymphocytes can make chemical messengers, and we're going to call them T helper cytokines. And so we're going to indicate them by this green arrow. The T helpers can make a great range of different cytokines. And the cytokines are used to activate the cells of the adaptive immune system. So T helper cytokines activate B lymphocytes, cytotoxic T lymphocytes, and will activate macrophages, right? So what I'd like to do just in the overview is to just walk you through what are the consequences of T helper cytokines activating B lymphocytes. Again, you guys, we just want to stress, why are these T helpers so crucial, right, to our adaptive immune responses? So with B lymphocytes, we already mentioned that B lymphocytes are going to make antibodies, right? We haven't yet talked about different classes of antibodies, but we'll just give you a little preview here. Okay, so when B lymphocytes first encounter an antigen, something they recognize as being foreign, they can start making antibodies, but they always start making one class of antibody called IgM. And IgM, again, don't mean any disrespect here, but IgM isn't the best antibody because it has a limited number of functions. It can't carry out all the functions we need to protect ourselves against invading pathogens. And furthermore, it's so big. It's like this, almost like a snowflake, like a five-part snowflake. It's so big, it often can't diffuse easily from inside our blood vessels into the extravascular spaces where a lot of pathogens can be. Another disadvantage of IgM is it can't cross the placenta. So if mom's making IgM against a pathogen, the IgM cannot cross the placenta, so the baby developing in utero can't acquire any of mom's maternal antibodies, and that's really important passive immunity for the baby. Because again, the baby's immune system, when the baby's born, is still underdeveloped, right? So when newborns first come into the world, they don't have really strong adaptive specific immune responses, so they're going to rely on mom's maternal antibodies to protect them. So how do the T helpers help these B lymphocytes? Well, in the presence of T helper cytokines, the B lymphocytes will undergo this amazing process called class switching. So in response to the T helper lymphocytes, the B lymphocytes can switch from making IgM to IgG. And we're going to say, you guys, IgG is the best antibody because it can provide all the protective functions of antibodies: neutralization, agglutination, opsonation, complement activation, and furthermore, you guys, it's IgG. IgG is the only antibody that can cross the placenta, right? So if mom's making lots of IgG against pathogens she's encountering, she can pass those to her baby developing in utero. So when the baby is born, the baby will have mom's protective circulating maternal antibodies on board. But again, you guys, B lymphocytes can't switch to IgG unless what? The T helper lymphocytes are there making the cytokines. And another really important role, folks, of the T helper cytokines is they will help trigger the B lymphocytes to some population to develop into B memory cells. So T helper and their cytokines are absolutely essential for the development of immunological memory. If there's no T helpers around, then the immune system can't trigger that secondary immune response. We won't get that faster, stronger, longer lasting, more effective secondary immune response if there aren't any T helper lymphocytes around. Okay, so that was T helpers and how their cytokines influence B lymphocytes to undergo class switching from IgM to IgG, and to make memory cells. So now you guys, let's go over here. So the T helpers will make, again, a variety of cytokines. So let's go over here to the side of the concept map that represents cell-mediated immunity. And let's first take a look at our cytotoxic T lymphocytes. So we'll see, folks, that cytotoxic T lymphocytes, they can recognize our cells that have been parasitized by intracellular parasites such as viruses, and using really specific surface receptors, they can bind to the surface of our cells that are displaying some of the viral antigen. And later we'll have a slide on this, you guys. And then what's so cool is then the cytotoxic T lymphocyte, it releases what some microbiologists call the kiss of death. Oh, I just love it. And what the kiss of death are these molecules. One of them is called perforin. The molecules will be released when membrane-bound vesicles chock full of these substances will fuse with the cytotoxic T lymphocyte membrane, and they will be dumped on the surface of our viral infected cell. Now, perforin is going to act a lot like the MAC attack of complement. So perforin, they're little proteins that will insert themselves into lipid bilayers, forming pores, perforation, right? And that can, if you have enough perforin, that will directly lyse the infected cell. And again, folks, another going back to the double-edged sword of our immune system. So that cytotoxic T lymphocytes will actually kill our cells. They're going to contribute to tissue damage. But remember, you guys, if we can kill our virus-infected cells early enough, the viruses can't replicate, right? And so then you stop the viruses from replicating, and you'll stop them from continuing to cause harm, right? Now, as always, there's always kind of like a 1-2 punch, it seems, in our immune system. So in addition to the release of perforin, another antimicrobial substance will be, not antimicrobial, excuse me, you guys, I said that wrong. Another cytotoxic substance will be released called granzyme. And what granzyme will do when it's released, it will enter our virus-infected cell through those little perforin holes, right? And the granzymes trigger a series of events that causes our cells to basically kill themselves, undergo programmed cell death, apoptosis, cell suicide. And again, the advantage of that is if we can do that early enough, the viruses can't replicate. So we can stop viral replication, we can stop the viruses from spreading to other cells and causing more harm. Okay, so that's, and again, what is required for cytotoxic T lymphocytes, right, to be activated so they can release the perforin and the granzyme? It's, this is going to be the default answer, you guys, T helper cytokines, right? That's going to be absolutely essential. And furthermore, the T helper cytokines are going to be essential for the cytotoxic T lymphocytes to make memory cells. Okay, so T helpers are essential in getting memory cells, memory B cells, and memory T cells to be made. Okay, and then our one last example, you guys, of how important T helpers are, we're going to take a look at a macrophage that's been parasitized by one of those facultative intracellular parasites, like good old Mycobacterium tuberculosis is kind of the poster microbe, is a facultative intracellular pathogen. So in a so-called resting macrophage or unactivated macrophage, the macrophage can ingest the Mycobacterium tuberculosis, but it can't kill it. Right? So what we need to do is activate that macrophage, turn it into an angry killer, right? And how are we going to do that? You got it, with T helper cytokines. And the one we'll mention, you guys, will be gamma interferon, the third kind of interferon. I always think of gamma interferon as high octane, highly caffeinated coffee for the macrophage, right? So in the presence of the gamma interferon produced by T helper lymphocytes, the macrophage is activated. It becomes a more efficient phagocyte. It's faster at phagocytizing, engulfing microbes. And very important, you guys, it's going to greatly increase the production of hydrolytic lysosomal enzymes, and it's also going to greatly increase production of those toxic reactive oxygen intermediates like superoxide anion and hydrogen peroxide and hypochlorite bleach-like substances, right? And again, you guys, we just want to emphasize that without the T helper cytokines, the macrophages can't be activated. So they end up being delivery trucks, right, for a lot of these facultative intracellular pathogens like Mycobacterium tuberculosis. It can spread throughout our bodies. So again, folks, we just want to come back to this. If you understand why it is that HIV can destroy our immune system, if you understand the key role of the T helpers, then you can understand how when HIV destroys enough of our T helpers, humoral immunity no longer functions and cell-mediated immunity no longer functions. And the tragedy is then that our patients that are suffering with AIDS, they can often die of maybe like an opportunistic pathogen infection, something that normally their immune system could totally control. So a lot of times, um, our AIDS patients, again, they'll die from opportunistic pathogen infections, or certainly if they do become infected with a highly virulent pathogen, it's it's just heartbreaking because they basically have no defenses, right? So again, you guys, if you get this big concept, if you don't learn anything else from adaptive specific immunity, you understand the key role of the T helper lymphocyte, I think you have the most important points. So the rest of this, we're just going to be filling in some details. All right, folks, so what triggers specific adaptive immunity? So anything that triggers a specific adaptive immune response is called an antigen. Now, antigen should be non-self, meaning it's a substance that your body makes shouldn't trigger a specific adaptive immune response. If it does, that causes a disease that we're seeing in increasing frequency called autoimmune diseases, right? So we want antigens, if our immune system is working properly, to be non-self, to be foreign, right? So the antigens are the triggers that tells our immune system we're being invaded, and we need to trigger these specific adaptive immune responses. Okay, so another way to say this, you guys, is antigens, for example, would trigger antibody production, right? And we would say, you guys, that if we took like a specific microbe, let's say maybe this is a bacterial pathogen here. We would say if the bacterial pathogen invaded us and triggered, say, antibody production, we would say that the bacterium is antigenic. But it turns out that different parts of the microbe might trigger different types of antibodies to be produced. So if we were being really, really accurate here, folks, I would be talking about um, um, antigenic determinants, or epitopes, are the specific parts of the antigen, and you guys pretend this is just some generic invading microbe. The microbe is an antigen. It's going to trigger, for example, antibody response. But the different parts of the microbe can trigger production of specific antibodies that will bind to that part. And I'll just make this up, you guys. So this might be, say, flagellar antigen. So the antigenic determinant, the epitope of the flagellum. So we're going to have one kind of antibody that will bind to the flagellar antigen. And again, you guys, it's good old lock and key, right? Or like enzyme substrate relationship here, right? And maybe over here, maybe this is a cell wall protein. So that the cell wall protein would act as a specific epitope or antigenic determinant. Will have a different type of antibody made that can bind to that shape, right? And maybe over here, maybe this is the conjugation pilus of the invading bacterium. And we'll see that then the pilin, the protein subunits, are the specific epitopes or antigenic determinants to which another type of antibody will bind, again, specifically based on complementary shapes, right? So folks, when I talk about, say, an antibody binding to the antigen, I should say, the antibody binds to the antigenic determinant or the epitope, but I usually don't. I usually just say the antibody binds to the antigen. So I just wanted to clear that up. And again, folks, um, in the concept map, we were talking about B lymphocytes and T lymphocytes and their ability to, we're going to see our B lymphocytes and T lymphocytes, they too can bind to antigens. And they're going to do it using cell surface receptors that function and maybe even look a little bit like antibodies, right? Okay, so just to kind of throw that in there. So folks, we're going to spend most of the slides talking about humoral immunity, the protection provided by antibodies or immunoglobulins, and this is to provide protection primarily against extracellular pathogens and toxins. Okay, and this is a cartoon, you guys, of a generic cartoon of an antibody, and this is the model we'll use to describe antibody or immunoglobulin structure. So, folks, again, if we were drawing up on the board, I would say this is an example of IgG. So IgG is a class of antibody that's in the highest concentrations in humans, and thank heavens, because it has the greatest variety of protective functions. So you guys, let's just presume this is an IgG. And we can see here, folks, that IgG, it is made up of 4 polypeptide chains. There's 2 short chains, this one and that one, and those are called the light chains. And then there's 2 long chains, this one and that one. Those are called the heavy chains, right? And so, oh, and this would be a great final exam question, you guys. So with an IgG antibody, it has primary level of structure, right? The specific amino acid sequence in the polypeptide chains. It would demonstrate secondary sequence with alpha helices and beta pleated sheets. It would have tertiary structure, right, folding into a complex 3-dimensional shape. But you guys, antibodies are an example of proteins that have quaternary level of structure, right? The specific arrangement of the polypeptide chains. And notice here, folks, the IgG quaternary structure, the polypeptide chains are arranged in this Y-shaped conformation. And notice, folks, that the light chains have very specific attachments to the arms of the heavy chains, right? So, for example, you guys, this antibody couldn't work if the light chains were attached down here to what we call the stem, right? Or if that light chain was attached down here to the stem, the antibody would be non-functional. So the antibody has to have the correct quaternary structure for it to function. Okay, so folks, again, just some terminology here. So you can see this looks like the letter Y, right? Or you guys, again, if we were in lecture, I'd be standing there with my arms stretched out over my head, and I would say, well, my arms are like these antibody arms. There's an arm there and there's an arm there. And at the tip of my arms, just like at the tip of the antibody arms, there's this very specific binding site. This would be like my hand, right? And it's at the tip of these arms, you guys, that the antibody binds to the complementary antigenic determinant or epitope. And we can see it right here, you guys. So here's our big antigen, here's the antigenic determinant or epitope, and this is the antigen binding site right here, right, at the tips of the arms. So folks, in a lab experiment, we could take a specific protease that can hydrolyze the peptide bonds here and the peptide bonds here, and that would result in 3 fragments of the antibody being released. Now, 2 of the fragments would be these arms, 1, 2, right? And these arms are called the FAB fragments. And F fragment, AB stands for antigen binding site, right? So experiments were done to prove that it's at the tips of the FAB fragments that antigens are bound. Now this stem down here, this is called the FC fragment, and the C is supposed to stand for crystallizable, but to me it's like that's not very helpful trying to learn its function. So you guys, so the FC stem is really important. It can bind complement and activate complement, so maybe you can think of SC, C is for complement binding. In addition, folks, this is really important, our phagocytic cells have receptors for the FC stem right here. Our phagocytes have receptors that can bind to the tip of the FC fragment. And we're going to see that will play a crucial role in the process of opsonization, where we're going to coat our invading pathogens with antibodies, and then that makes it easier for our phagocytes to attach and ingest them and kill them. Okay, just a little bit more detail, you guys. So we'll you'll see this lighter shaded portion of the heavy chains and the light chains here. These are called the constant regions. So for any antibody class, for example IgG, all the different IgGs would have the same amino acid sequence here in the constant regions, right? But then at the tips of the FAB fragments, you can see this is called the variable region. So if we had 5 different IgG antibodies that bind to 5 different antigenic determinants or epitopes, the amino acid sequence here in the variable region would all be different. And this makes sense, you guys, because remember, it's the antigen binding site is at the tip of the FAB fragments, so the amino acid sequence at the tips will have to be different to make the different complementary antigen binding sites here. Okay, and let's see here, folks. So furthermore, oh, and I forgot, and I apologize, you guys, when we were talking about the levels of protein structure, in this simplified cartoon, folks, we're not seeing like the alpha helices, the beta pleated sheets. We're not seeing all the R group water interactions that we would see that stabilize tertiary and quaternary structure. But what they are showing us, folks, here are disulfide bridges, right? So that's the strongest R group interaction. So usually they only show the disulfide bridges here, but again, there's going to be a lot more R group and R group water interactions stabilizing tertiary and quaternary structure. Okay, I think that's it for this little guy. So then on to functions of antibodies. And you guys, it's so good because we've already covered some of this in the immune responses to viruses. So because if, again, you guys, if we use IgG, the predominant antibody, you'll remember it has two FAB fragments and 2 antigen binding sites. So that means a single IgG can bind to 2 separate pathogens, right? And so if you have a number of pathogens and you have IgG antibodies, they can cross-link the pathogens together, forming these great big clumps. So that process where the antibodies cause clumping, it's called agglutination. So these antibodies would be called agglutinating antibodies. Now, why is that helpful? Well, by causing the pathogens to clump together, it's going to make it much harder for them to spread through neighboring tissues. And also, folks describe that it's easier for phagocytes to phagocytize a number of pathogens at the same time if they're in these clumps. And then, folks, this is another protective function of antibodies that we've talked about, neutralization. So remember, neutralization occurs when the antibodies bind to the adhesins on the surface of the pathogen or the portion of, say, an exotoxin that binds to our cells before it can get into our cells. And so neutralizing antibodies, you guys, are so wonderfully protective, you know, and this is the big debate that's going on right now in folks that have survived infection with the pandemic coronavirus. They have circulating antibodies, but I don't think folks know yet, or there isn't an easy test to see, you know, are those antibodies that you have against the pandemic coronavirus, are they neutralizing, right? Basically, are they protective? So there's just so much more we need to know. But again, folks, we just want to remember that if we can block attachment of an exotoxin to our cells, or if we can block attachment of a pathogen to our cells, there won't be any harm, right? You're blocking that first crucial step. If there's no attachment, then usually there's no harm. So we really need, I know you guys are tired of me saying this, we really need a pandemic coronavirus vaccine, and it would just be awesome if it would trigger, you know, huge amounts of neutralizing antibodies. Okay, so here again, neutralizing antibodies against an exotoxin. Here's neutralizing antibodies against a virus, so we see that antibodies are attaching to the adhesins, and likewise here with this invading bacterium, going to block attachment. Down here, folks, again, I think this is a concept we introduced already. Here's our phagocyte, right? And remember, in phagocytosis, the phagocyte has to attach to the surface of the invading pathogen, right? And you'll recall there is a problem because our phagocytes have never evolved receptors for the capsular polysaccharide. So encapsulated bacteria, they just can escape from our phagocytes, right? So that's such a bummer. So a couple of strategies have evolved in general to increase phagocytic killing, and the process is called opsonization, and it literally means preparing to eat. So I think previously, you guys, we had mentioned that with complement activation, if complement can coat the invading pathogen, our phagocytic cells have complement receptors, right? So a phagocyte can much more efficiently ingest and kill a complement opsonized bacterium, right? And furthermore, you guys, again, that good old 1-2 punch, antibodies are also opsonic. And again, you guys, the best class, we'll be talking about classes of antibodies, the class of antibodies that are the best opsonic antibodies are the IgG, right? Good old IgG. And the reason that IgG is opsonic is we can see here, if this is a single IgG, it's attaching to the pathogen at the tips of the FAB fragments at antigen binding sites, but that means the FC tip is free, right? And what do phagocytes have? They have FC receptors here, right? So if we can coat the invading pathogen with IgG, then the phagocytes can bind to the FC tips with their FC receptors and much more rapidly ingest and kill the invading pathogens, right? So those would be opsonizing antibodies. And folks, this is just a little cartoon to, again, talking about the 1-2 punch of the immune system, right? So we said that not only is complement opsonic, but antibodies are opsonic. So let's pretend this is an invading microbe and it's been opsonized with both antibodies and with complement. So we'll just make this up, you guys. So here's the opsonized bacterium. So let's say here on the phagocyte, maybe this is the receptor for complement, right? So it binds a complement that's opsonizing the bacterium. And let's say this is the receptor for that FC tip of the FC fragment, right? So if you have both complement and antibody opsonizing your invading pathogens, that hugely increases phagocytic killing. And just some terms, you guys, so the opsins tag the invading microbe, it makes it stickier, right? So it's easier for them to be phagocytized and destroyed. I think of opsins, you guys, because I'm always thinking about food. I think of opsin as like sticky chocolate sauce that we're going to pour over this microbe and make it much stickier and more delicious for the phagocytes to kill. So again, folks, this is just another concept map going over antibody function. So we've talked about neutralization, agglutination. If you have soluble antigens, like maybe exotoxins, you can actually precipitate the exotoxin with precipitating antibodies. And then all of these, in this little cartoon, you guys, it's showing that that the antibodies again can increase phagocytosis, right? If the phagocytes have receptors for them. And then, folks, this is the reason I actually put this concept map in here. Is another cool protective function of antibody is that some antibodies, when they bind to their antigen, so let's say this is an invading microbe of some kind, right? So when the antibodies bind to the microbial antigens, there's a little change in the shape of the FC fragment, and that permits a complement protein to bind and get activated. And you guys remember, with complement activation, all of the advantages. We make inflammatory mediators, we make chemotactic factors, we make more opsonins, right? And very importantly, we're going to make those membrane attack complexes, right? The activated complement proteins insert into the lipid bilayers, causing holes, right? So that can cause direct lysis of cellular organisms. The MAC attack can damage the envelope of viruses, and we know if the viral envelope is damaged, they're no longer infectious, right? We can get holes formed in the outer membrane of gram-negative bacteria, and then the complement can get into that periplasmic space and then insert into the cell membrane of the gram-negative bacteria, leading to lysis, right? So these antibodies are just awesome. So folks, we're going to do this ever so briefly, the classes of antibodies. So again, going back, you guys, on our immunology module, there was the scan of a couple of pages out of an immunology book. So on the first page, you guys, it's page 523. Down in the lower right hand corner, there's table 61-1. It says important functions of immunoglobulins. So these are really brief descriptions of the antibody function, but I'll try to verbally tell you what I'd really like you to know for our final exam. So let's start, folks, first with the first antibody that B lymphocytes make, and you might remember that's called IgM, right? And if you look at IgM, it looks almost like it's almost like a snowflake made out of 1, 2, 3, 4, 5 kind of basic antibody molecules, right? So IgM is often referred to as a pentamer, and see how big it is, you guys. This is a huge antibody. And you will recall, you guys, we said IgM, it's so big, it usually doesn't diffuse out of the blood vessels into the extravascular tissues. So it's not in really high concentration in extravascular tissues. And furthermore, you guys, mom can't pass her maternal IgM across the placenta, right? So there's no transplacental transfer of IgM. Now, IgM, it's an awesome agglutinating antibody because look, you guys, how many antigen binding sites are there? There's 10, right? So a single IgM could cross-link 10 invading pathogens. Fantastic agglutinating antibody. But is a pentamer, you guys, it's not opsonic, right? Because see how the Fc tips are pointed inward, so the phagocytes can't get in there to bind to them, right? So not the best, definitely not a great opsonic antibody. Um, it can fix complement. It can fix complement, so that would be a good thing, right? But again, you guys, in relatively low levels in the extravascular tissues, it can't cross the placenta. It's really not a great opsonic antibody. So, I mean, it's helpful, but not the best. So what's the best antibody that can do everything? It's this one, IgG. And I think, you guys, there's been natural selection that IgG is the predominant antibody in most of us. I think it represents about 80% of all of our antibodies under just normal situations. And the reason it's so fantastic is, let's go through all of its protective functions. It's neutralizing, it can agglutinate, it can precipitate, it can opsonize, it can activate complement, and you guys, this is the only antibody that can cross the placenta, right? So if mom's pregnant, she's passing her protective maternal IgG across the placenta to her baby. So when the baby's born, baby already has mom's circulating protective IgG antibodies protecting the baby against all the pathogen that mom encounters day to day in her environment. Isn't that just amazing? I just think that's phenomenal. Okay, so if I ask you, what's the best antibody of all the classes, the antibody that offers the greatest number of protective functions, it's going to be IgG. And remember, you guys, B lymphocytes, they don't do they don't do good class switching um, unless T helper cytokines are available. So if all the T helpers were gone, the B lymphocytes, they would just get stuck making IgM. And again, not the best antibody. And then, folks, we're going to go over here. This is another amazing antibody. So we can see it starts out as it looks a little bit like an IgG, right? But these antibodies, they get secreted across the epithelial cells of mucous membranes. And when they're secreted, they have this a J chain and a secretory component attached. And the J chain, sorry, is going to attach two of the monomers together to make a dimer. So these IgA dimers are called secretory IgA, and we're going to find them in really high concentration in that mucus blanket, right, of our mucous membranes. And you guys, it's also really rich in breast milk. Yeah, this is so cool. So we can see, folks, with a secretory IgA that's going to be rich on all of our mucous membranes. They can be neutralizing, right? Can be neutralizing, they can block attachment. And furthermore, you guys, when they attach to, say, a toxin or a pathogen, the secretory IgA will get bound to components in the mucus blanket, right? And remember, you guys, all those mucus blankets, they're all eventually removed from our body. You know, it doesn't matter if it's our conjunctiva or respiratory tract or gastrointestinal tract or genital tract, that mucus blanket is always eventually going to get removed from the body. So if the secretory IgA is bound to a pathogen and then it gets literally bound to the mucus blanket, that will guarantee that the pathogen or the toxin is excreted, removed from the body. I just think that's amazing. And again, folks, talking about passive immunity offered by mom's maternal antibodies, breast milk in mammals is chock full of secretory IgA. And so this is fantastic, you guys. So if a baby is nursing, being breastfed, the baby's intestinal tract is being constantly showered in mom's protective secretory IgA. So if the baby, is, I don't know, babies love to put everything in their mouth, right? And can you imagine all the microbes they're ingesting when they're putting everything in their mouth? So as long as the baby is breastfeeding and has mom's secretory IgA on board, the maternal secretory IgA can bind to those pathogens, right? And prevent them from binding to the baby's intestinal mucosa, and make sure that those IgA-coated pathogens then will just get pooped out, right? And that's exactly what we want to um, have 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 happen to those pathogens, right? So again, folks, if mom can, it's so good. If mom can breastfeed, there's a lot of conditions where moms can't. There could be medical conditions, maybe just circumstantial um, um, situations, right? But you guys, formulas, huge, kind of worldwide, huge issues with baby formula, infant formula, right? So number one, it is so expensive. It's so expensive. So that's a great hardship on families if they're having to buy infant formula. And furthermore, you guys, in many parts of the world, the water that's used to reconstitute the powdered infant formula is not clean, right? So in some parts of the world, if you have fecal contaminated water and you're using that, right, to make the infant's formula, then your baby's going to be like doubly at risk for diarrheal disease. Yeah, it's really, really cool, you guys, because they have breast milk banks, right? Where women who are making, extra breast milk, they can donate the breast milk to these breast milk banks, so that babies that are in need, of human breast milk, can have access. And one last plug, you guys, for breast milk is, as mammals, the first breast milk that mom mammal makes right after giving birth, it's called colostrum, and it's thick, chock full of protective mom's antibodies. So if possible, if there is a situation where a mom can't breastfeed, if she could at least get that colostrum to her baby, that's going to get that baby off to a great start in life, protecting the baby against all of the possible intestinal pathogens the baby might encounter. And again, you guys, in the breast milk banks, they have colostrum banks, right? So if there's a medical situation where the mom can't provide colostrum to the baby, we can have donations of colostrum from the breast milk bank. So I think that's just fantastic, you guys. All right, and then you guys okay, almost finished here. So let's go down to IgE. So IgE probably originally evolved to protect us from large parasites, like eukaryotic parasites like worms. Just think, you guys, of like an ascaris worm in your intestine. That's way too big for your phagocytes to kill. Or let's say the microfilaria, right, of Wucheria bancrofti. Our phagocytes are too tiny to be able to ingest them. So one of the functions of IgE is that it can bind to certain cells of our immune system, and I believe it's the eosinophils that it can bind to by the Fc tip. And then if you have an eosinophil that's kind of loaded, armed with these IgE, then the IgE can bind to the large parasitic worm, and that will trigger the eosinophils to release these anti-parasite proteins. The functions kind of are like perforin, right? That we mentioned earlier with the cytotoxic T lymphocytes, right? So again, IgE, and sometimes in parasitic infections, you'll see, if we're talking about eukaryotic parasites, you might see an elevation in IgE antibodies, which is kind of interesting. Nowadays, in areas of the world where, people almost say like we've almost become too hygienic, because in areas of the world where we aren't seeing heavy parasite loads, right, this IgE is starting to get into mischief. So IgE can bind to mast cells, again, by this Fc tip, right? So if you have loaded like armed mast cells with IgE on the surface, the problem is, if you're exposed to the antigen to which the IgE binds, it triggers the mast cell to undergo a process called degranulation. And when mast cells degranulate, they release huge amounts of inflammatory mediators. And the classic one is histamine, right? So you guys, so the IgE in mast cells, they play an important role in type 1 hypersensitivity reactions, anaphylaxis. Now, if this type 1 hypersensitivity occurs just locally, like say in your nose, right? Maybe you know, the so-called hay fever, you have an allergy to maybe plant pollen. If it just happens locally in your nose, it's not life-threatening. You're uncomfortable, your mucous membranes become swollen, it's hard to breathe and maybe a lot of mucus, right? But it's not life-threatening. But the problem is, you guys, if you have lots and lots of your mast cells armed with these IgE, and then you get exposed to the antigen, which we will now call an allergen, you can get massive release of those inflammatory mediators, kind of like the equivalent of that cytokine storm, and that can throw a person into life-threatening anaphylaxis, right? So an example, you guys, in comparing hay fever in the upper respiratory tract, relatively mild, local, if you have that reaction in the lower respiratory tract, right, then you're talking about asthma, and that can close down the airways, right? So asthma can be life-threatening. The peanut allergies that we're talking about, drug allergies, right? Those types of allergies, if you get enough mast cells releasing those inflammatory mediators, they can be life-threatening, right? So it's so interesting that IgE, it evolved to protect us against large parasites, eukaryotic organisms, and now we're not seeing so many of those, so now it's kind of getting into mischief. That's not very scientific, is it? Okay, and then folks, this last one, IgD, they find it is a surface receptor on B lymphocytes. I think there's still, kind of a question as to why the D, but it can act as one of the surface receptors on B lymphocytes. It will bind to antigens, right, and help to activate the B lymphocytes. So again, folks, this would be important to know for our final exam. If I ask you which cells make antibodies or immunoglobulins, it's the B lymphocytes. And once they start making antibodies, they are awarded a new name. They're called plasma cells. So a plasma cell is just a B lymphocyte that's making antibody. So folks, in our bodies, we have a number of B lymphocytes, right, that have these surface receptors. And again, you guys, some of these surface receptors, they could be IgD. And notice, you guys, how the surface receptor, it looks like an antibody that's binding by the Fc end here, right? And if we looked at all the, thousands of different lymphocytes circulating in our body, we'd see that they can all bind to different antigenic determinants, different epitopes. So you guys, here they've just colored. So we'll just make this up, you guys. Let's say this B lymphocyte can bind to antigen A, and this lymphocyte can bind to antigen B, and this lymphocyte can bind to antigen C. And all those antigens, all those epitopes, they have different shapes, right? So we want to know, well, how does the correct lymphocyte get activated, right, to make antibodies against an invading pathogen? So it turns out, you guys, it's the antigen, or specifically antigenic determinant or epitope, that selects the right B lymphocyte. And the way the antigen is going to select the correct B lymphocyte is by binding to the surface receptors, right? And again, you guys, it's kind of like the lock and key or enzyme substrate. The antigen has to have the correct shape, size, and charge distribution to be able to bind to the surface receptor of the B lymphocyte. And here, folks, like a drawback of this cartoon is we're not seeing that T helpers have to be present here. The T helpers have to be providing those T helper cytokines for the next events to occur. So you just kind of have to imagine the T helper is always there helping. Okay, so you guys, so we would say then the antigen selects which B lymphocyte will be activated. So antigen binding is going to help activate the lymphocyte, and as part of the activation, you guys, the lymphocyte is going to start dividing like crazy. It's going to proliferate, right? So it's dividing like crazy. So these are like little genetic clones of itself, right? So so the first lymphocyte divides in 2, and they divide in 4, and on and on and on. Now notice, you guys, that is part of this cool process of clonal selection and activation. Notice that there's going to be 2 subgroups of clones. This group right here are the plasma cells, and they're going to make antibody. And here you guys, initially it would be IgM, and then with T helpers again, they're going to switch to IgG. So they're kind of showing us, they kind of skipped a step here, but that's okay. But then you guys, here is key to the memory of specific adaptive immunity. A second subpopulation of those little clones, these little guys are going to be the memory B cells. They're not going to make antibody. Instead, their job is to live long and remember that antigen that triggered this initial activation. So these memory cells, they're like pre-primed. They can live months, maybe years, right? They're just waiting. They're the memory of the immune system. So then subsequently, months or years later, if that same antigen, the same antigenic determinant or epitope is encountered, it binds to the memory B cells. And like we said, you guys, these guys are pre-primed. They start dividing like crazy. They can initiate antibody production and antibody class switching much more rapidly, right? We're gonna get more plasma cells made, and furthermore, you guys, in that secondary immune response, right? We're going to get even more memory cells made, right? So you guys, this would be the first time the lymphocytes encounter the antigen. This is a primary immune response. We're going to see that immune response tends to be slow and low, not very effective, not very long lasting, but we make memory cells in the primary immune response. And then it's those memory cells that will trigger the secondary immune response, where we're going to get a faster, stronger, longer lasting, more effective, higher IgG concentration in the secondary immune response than the primary immune response. And that's what would provide, in theory, lifelong protection, lifelong immunity against a pathogen. And it's also, you guys, the basis of vaccination, right? When we vaccinate, we're trying to trigger this primary immune response, so we'll get those memory cells made, right? And then if we encounter the real pathogen or the real exotoxin later on, these memory cells will become quickly activated, and through really fast IgG production will hopefully protect us from serious harm, right? And again, we're going to make more memory cells. Just fascinating. And again, folks, just want to emphasize that these memory B cells, they can't be made unless who's present? The T helpers, right? Making the T helper cytokines. Okay. And this, again, folks, I know graphs are kind of, you know, in lecture, this, you guys would probably be snoozing by now if you throw up a graph, right? After 60 minutes of lecture. But you guys, these are really cool graphs. So what we're doing here is we're comparing the primary immune response, the first time the antigen is encountered, and we're comparing specifically antibody production here, and the primary immune response, we're comparing it to the secondary immune response, right? So let's take a look, you guys, at the primary immune response. So this is, I love this, you guys, because here they're using tetanus toxoid vaccination, yeah? So remember, the tetanus toxoid is a tetanus exotoxin that's been modified so it can't cause harm, but it'll still trigger a protective immune response. So you guys, these are some of the disadvantages of the primary immune response. Look at this lag period, right? The response is relatively low. It's almost 2 weeks before we get a peak of our good multifunctional IgG, right? We could die in that 2 weeks. But remember, you guys, innate nonspecific defenses are trying to protect us during this period until these big guns can be activated. Notice, you guys, here that the first antibody that's made is IgM, and then eventually with T helpers we're going to switch over to IgG. But notice here, you guys, it's slow. Antibody concentrations are low, slow, low. Not a lot of IgG, right? And it's relatively short-lived, yeah? But what's the beauty of it? Look at these guys down here. This is the beauty of the primary immune response, right? We're going to make those memory B cells. They're going to live long and remember that antigen, right? So if within 10 years, right, if we step on that rusty nail and jam those Clostridium tetani endospores into our foot and they germinate and they start pumping out tetanus toxin, right, the tetanus toxin is going to bind to those memory cells. The memory cells are pre-primed. The memory cells are going to start dividing like crazy, right? And because they're pre-primed with T helper cytokines around, they're going to rapidly switch over to IgG production, right? The best antibody of all. So Classic exam question, you guys, is comparing the secondary immune response to the primary immune response. So the secondary immune response is going to be faster. It's going to be stronger, meaning higher antibody production, much higher and faster IgG production. This is what we really want, right? It's much longer lived, right? The antibody production goes on much longer. And what else? We're going to get even more memory cells, yeah? So again, the basis, you guys, of vaccination and the basis of, like, if we say in the old days, if somebody survived smallpox, we said they would have lifelong immunity, and that was because of these beautiful memory cells. Secondary immune response, you guys, I'm just going over this and over this. Remember, we can't have the secondary immune response unless the T helpers were present in the primary immune response. So folks, this is like a little bit complicated, but to me it's also really awesome because it shows that humans can be really creative and come up with some wonderful strategies to help protect us and our children from some of these bacterial pathogens that have some pretty sneaky virulence factors. So a lot of this is just going over vocabulary and concepts, you guys. So so first of all, remember how I keep saying the T helpers have to be present. We need T helper cytokines, right, to make the memory cells. We need T helper cytokines to get the B lymphocytes to switch from IgM to IgG. Well, in all of those discussions, I didn't tell you this, but I was describing what are called T-dependent antigens. T-dependent antigens were triggering the immune response that recruited the T helper lymphocytes to get involved. So you guys, we would say T-dependent antigens, T helper dependent antigens are the best antigens, right? Because they're gonna recruit the T helpers that will trigger class switching from IgM to IgG. They are essential, the T helper cytokines are essential for making the memory B cells, right? So without the T helper cytokines, there's no memory. And another thing, you guys, that's really important with T-dependent antigens, they're really good antigens. And so little children and older adults who often, their immune system in little children, babies, it takes a while for the immune system to develop. And as older folks, our immune system is starting to fail us, right? So we have a hard time mounting a good immune response against some antigens, against some pathogens. But these T-dependent antigens, these are the best antigens to get little kids, babies, us old folks to make a protective immune response. So T-dependent antigens, you guys are awesome. So T-dependent antigens are usually proteins. Usually proteins. Proteins are the best antigens, right? Because the protein antigens are T-dependent. They're going to recruit the T helpers, and the T helpers are going to supply all those important cytokines for IgM to IgG class switching, for making memory cells, and they're also the ones that might trigger us, the really young, the really old, to mount a protective immune response. But the bad news is, you guys, there's other microbial substances out there, right? And many of those microbial substances, especially polysaccharides made from repeating monosaccharides, right? So like the same sugar over and over and over again. Those antigens are called T-independent because they don't get the T helpers involved, right? And if you can think of this, you guys, and again, if we had the whiteboard, I could draw on it, but guys, think of the capsules of bacteria. They're usually made out of polysaccharides, right? And the polysaccharides are usually the same monosaccharide, the same sugar unit linked together over and over and over again. So like the proverbial pearls on a necklace. And what can happen with those capsular polysaccharides, those T-independent antigens, like a single strand of polysaccharide can bind to several receptors on the same B lymphocyte, cross-linking the receptors, and that will trigger the B lymphocyte to start making IgM, but no T helpers have gotten involved, right? So that B lymphocyte, it's just stuck making IgM. And what else can't it do? The B lymphocyte can't make memory cells, right? The capsular polysaccharides, you know, the very old, the very young, we usually don't mount a very good immune response against them. So this is huge, you guys, because remember all those encapsulated bacterial pathogens like Streptococcus pneumonia and Haemophilus influenza and Neisseria meningitidis. These are all encapsulated bacterial pathogens which might initially start out as a respiratory tract infection, but they can invade into the bloodstream. And because of their capsules, right, our phagocytes, our macrophages, our neutrophils, they have no receptors. So those encapsulated bacteria, they spread throughout the body, they can invade the central nervous system, and they can cause fatal infections, right? So for so many years, people were trying to make vaccines against them. But it would make sense, right? You'd want a vaccine that would trigger opsonizing antibodies against the capsule, right? Because you'd like to have a vaccine that would trigger antibodies that could opsonize the encapsulated bacteria, maybe neutralize them, right? But the problem is, you guys, if you take the capsular polysaccharides, they're T-independent antigens, right? You're not going to get, all you're going to get is IgM, no switching to IgG, and furthermore, no memory cells, right? So what's a vaccine if there's no memory cells? So, and again, folks, this is just kind of the key point. So that these T-independent antigens, the capsular polysaccharides as an example, they activate the B lymphocytes without T helper cytokines. So the B lymphocytes can only make IgM and not for very long. There's no memory cells made, and babies and us very old folks, we can't respond well to these antigens. So this was a dilemma, you guys, because we need vaccines against strep pneumonia, Neisseria meningitidis, and Haemophilus influenza capsular type B. So you guys, this was brilliant. So what the folks developed is what are called conjugate vaccines. And this is so cool, you guys. What they do is they take the capsular polysaccharides from these pathogens and they link them to protein, right? So now the conjugate vaccine is protein linked to the capsular polysaccharide. This then is injected as a conjugate vaccine. And what's so amazing, you guys, is linking the protein to the capsular polysaccharide, the carbohydrate. Now T helpers are recruited. You've turned your capsular polysaccharide into a T-dependent antigen, right? And you guys, this is so fantastic. This has saved so many lives. So now we have conjugate vaccines for you young folks for bacterial meningitis. There's a conjugate vaccine to protect you against Neisseria meningitidis. For little babies, the Hib vaccine, that protects babies against Haemophilus influenza capsular type B. And for us old folks, we have conjugate vaccines to protect us against Streptococcus pneumonia. And again, you guys, in us old folks, one of the serious consequences of us having influenza, if we survive the influenza viral infection, often we end up with a secondary Streptococcus pneumonia that can kill us, right? So having those conjugate vaccines is just fantastic, you guys. It's such good news. Good news. We need more good news, right? Okay. So now, folks, just really quickly, we're going to blast through CMI, cell-mediated immunity. And you will recall, you guys, we said that it provides protection against intracellular pathogens because antibodies can't get into our cells, so we have to have another strategy. And in addition, CMI is involved in protecting against tumor cells, cancer cells, large parasites, and transplanted tissues. We're just going to stay focused on the microbes. So you guys, you might recall in that concept map at the very start, we have two examples: activation of macrophages by T helper lymphocytes, right? And that helps the macrophages to kill the facultative intracellular pathogens. And then the other example, you guys, are cytotoxic T lymphocytes. So we have a big T for the T lymphocyte and then a C for cytotoxic. These are also called CD8 positive T lymphocytes. How they're going to kill, how the cytotoxic T lymphocytes will kill our cells that have been parasitized by intracellular parasites like viruses. So folks, I apologize, it would be nice to have a nicer diagram here, but this is mostly just text. So in activation of the macrophages, again, who is the hero here? It's the T helpers, right? Our CD4 positive T helper lymphocytes. They're going to supply the cytokines, that gamma interferon, that will activate the macrophages to turn them into angry killers. And you'll recall, you guys, we use the example of a macrophage being parasitized by Mycobacterium tuberculosis, right? If the macrophage isn't activated, if it's a so-called resting macrophage, the Mycobacterium tuberculosis is not killed inside and can replicate inside the resting macrophage. So the T helpers are recruited. They're going to secrete gamma interferon. The gamma interferon, you guys, it's like that high caffeine coffee. That's going to wake up that macrophage, activate it, changing it into an angry killer. So some of the changes we'll see in those activated macrophages, they'll be faster at chemotaxis. They're literally moving faster. They're eating faster. They increase phagocytosis, the ingestion of the invading microbes. And here's really important, you guys, are going to increase production of those hydrolytic lysosomal enzymes and those toxic reactive oxygen intermediates like superoxide anions, hydrogen peroxide, and the bleach-like hypochlorite substances, you know, the old 1-2 punch. And the key is, you guys, we have to get those macrophages activated early enough, right, to destroy the mycobacterium early in infection. And again, folks, the bad news is in babies, little kids, us old folks, in our HIV/AIDS patients, this activation often does not occur rapidly enough, right? And so tuberculosis can be devastating. And then, folks, this is I think the last slide here. The remainder of the slides, you guys, we aren't going to discuss. They're only for your interest. And the other slides, you guys, I'm pretty sure those are topics that will be covered in your A&P class. So, you guys, this is the second example of cell-mediated immunity. And again, folks, what's not shown in this cartoon is we have to have T helpers here present, providing those cytokines to activate our cytotoxic T lymphocyte. So here's our cytotoxic T lymphocyte, T is for lymphocyte, C is for cytotoxic. These are also called CD8 positive cells, right? And in this story, you guys, this is one of our cells that's been invaded by a virus, right? So our cells have the capacity to take some of the viral proteins and present the viral proteins on the surface of our cell on what I think of as a serving platter, right? And you guys don't need to remember this, but these serving platters that all of our nucleated cells have, they're called MHC class 1 molecules. So this is a serving platter. There's a viral antigen that's on the serving platter. So this cell is presenting the antigen for neighboring T lymphocytes. This cell is saying, I'm infected, you need to kill me. Kind of like in the, oh my God, not the Alien movie. Oh, I'm forgetting the name of the great movie. Oh gosh, okay. Need to go back and review my movies involving parasites inside of people. So this cell is signaling cytotoxic T lymphocytes, I'm infected, you need to kill me. So here we see the cytotoxic T lymphocyte that has the complementary receptor to that antigen being presented on the MHC class 1. Here we see the role, you guys, of that CD8 molecule. It's stabilizing this interaction so the cytotoxic T lymphocyte can get activated. Again, the second signal, first signal antigen binding, second signal is T helper cytokines. And so here's what the cytotoxic T lymphocyte is going to do, you guys. So here's the infected cell. Here's these membrane-bound vesicles chock full of perforin. The vesicles are going to fuse with the T lymphocyte cell membrane, dumping the perforin into that little tiny space. The perforin is going to insert into the cell membrane of the infected cell, right? And if we have enough perforin, the cell's going to lyse and die, and that'll stop replication of the virus. That's what we want, right? And then again, remember, you guys, there's always a 1-2 punch. So if the perforin doesn't do it, here's another cytotoxic chemical granzyme. So the granzyme will be released. It's going to enter the infected cell through the perforins, and the granzyme is going to trigger activation of the enzymes involved in apoptosis, the programmed cell death or cell suicide. And as a consequence, this cell will die, right? And we want that. It's like it's our own cell. Why do we want that to happen? Because we want to stop the replication of this virus. If the cytotoxic T lymphocytes can kill our cells that are first infected early in the viral infection, you stop viral replication, right? And therefore you stop the viruses from causing further damage to us. So you guys, again, I love this. This is often called the kiss of death, right? The perforin and granzymes, the kiss of death. And folks, I think that's it. Right. The other slides, you guys, we always, or at least I do, we always run out of time to go through these details. So don't worry about them. Again, you're going to get fantastic immunology in A&P. And gosh, you guys, that's our last lecture. This has been a wild semester. I wanted to thank all of you for your incredible patience, your good humor, your adaptability, right? It's just been phenomenal. And I'm so sorry that I don't get to say goodbye to you and wish you all the very best in person. Um, I'm hoping as soon as they can get a vaccine, right? You guys, life will return to normal. So hopefully we'll have a vaccine here. Hopefully, I sure hope January of 2021. But I just want you guys to know how much I appreciate you at so many different levels, and I appreciate those of you that are going to be um, um, pursuing careers that are going to help other people. I just applaud you so much. All right, you guys, and just really briefly, we've got our comprehensive final exam that's coming up not next week, but the week after that. So previous semesters, students have said they've just used their lecture study guides to get ready for the final exam. What I thought I could do, there's no way, you guys, I can do a comprehensive review because for the final exam because it's the whole semester. But what I thought I could do is maybe put a PowerPoint together that just has a few select slides from each of our lecture units, and then I could just do an audio just like this. And again, you guys, it can't be comprehensive, right? But I can bring up to you some themes, right, that are emphasized on the final exam. I'm hoping that I can make the final exam so it does include a lot of clinical, kind of clinical or environmental microbiology application questions where you're integrating a lot of the information from the different lecture units and hopefully making it a little bit more real world. And maybe in that little short PowerPoint, some key points. Maybe I could give you some examples. Okay, you guys, so take good care, stay healthy, and thinking about you all. And I know I'm going to see you one of these days. Okay, take care, you guys.